to get started? Okay, well, off you go and introduce yourself.
Well, thank you very much. I'm Richard Rainbow. I'm a relatively new recruit to the University of Liverpool. Our lab group moved here in May of last year. Moved here from the University of Leicester. And yeah, so we're now, originally I moved to be in the Department of Pharmacology, but now with the Project SHAPE, we've moved into the Department of Cardiovascular and Metabolic Medicine. I'm going to talk today about a project that we've been working on for quite some time now in collaboration with Caroline Dart and Richard Barrett-Jolly as well more recently, looking at the function of a channel in the heart that we kind of have overlooked in the past, and it's something that we think is actually a really important part of normal cardiac regulation. So I'm going to start off by giving quite a lot of background slides to try and introduce where we're coming from with this work. But first thing I wanted to talk about was something really quite basic, is about what causes damage to the myocardium during myocardial infarction or a heart attack. Now, we all know that you block a coronary artery and that stops the perfusion into the muscle. So if we have a coronary artery block, say at this point here, all of the downstream tissue from that artery is going to be poorly perfused during that time. So that's tissue ischemia, where you've got a lack of blood flow or a severe reduction in the blood flow. And the damage imparted during this ischemic insult not only happens during the ischemia, but on reperfusion, you can still have some problems with the microvasculature, so they don't open up properly after that reperfusion. So actually ischemia can carry on for a lot longer than just during the main period of ischemia. So when that ischemia is relieved, so the block's taken away, and that could be due to removal of the clot by balloon angioplasty or by clot busting drugs, the cells there can be actually more damage that occurs during that reperfusion phase. And that occurs due to the fact that during the ischemia, you've had lots of calcium build up inside the cells, so you get this calcium overload. On reperfusion, if you suddenly give back all of the oxygen and the substrates and things that the muscle needs to contract, and you've also got high calcium, you can get this really severe contraction, often referred to as hypercontracture. And that can, in some cases, physically damage the cells. You can actually rip holes in the membrane, and that obviously kills the cells. Further to that, you also get necrotic cell death from the really high calcium inside the cell. That can lead to further apoptotic death around it, and so your infarct gets bigger, the area of damage to the heart gets bigger. So the key mechanisms really of ischemic injury are before you have a coronary intervention and following coronary intervention again. So during your ischemia, if this is your blood vessel and this is a clot in the blood vessel, anything that's downstream of that block is going to be ischemia. It's got no blood flow, so that's where that damage can occur. And then again, once you've removed that clot and you put a stent in place, occasionally bits of that clot can come off and get stuck in these smaller vessels, or generally the vessel can just be contracted because of the fact it's had ischemia, and it can cause release of local contractile metabolites. And that means that you can't reperfuse that area properly. So although you've reperfused the main vessel and most of the tissues being reperfused normally, some of it can still remain ischemic. So ischemia can last a lot longer than just during the blocked artery phase. So there's a huge unmet clinical need with heart attacks, and this is one of those classic slides that we always have to do when we give translational type talks, that in the UK alone, there's around 85,000 heart attacks a year, and that gives us a cost of about 2.5 billion to the NHS. And our main treatment strategies still remain opening up the artery by either balloon angioplasty and then putting a stent in place to hold it open, or in patients that you can't do the balloon angioplasty because they might be unstable, or it's going to take too long to get them to the clinic to have this, you can give them thrombolysis, these clot busting drugs. So with all of these things, there's an inevitable delay in getting that patient to the hospital to have the procedure, or getting in the thrombolytics to break that clot down to reperfuse. And again, this poor perfusion might carry on for up to 48 hours due to this microvascular dysfunction. So really there's this urgent unmet clinical need to find something new in our treatment strategies for infarction to try and address the ischemic damage that occurs during and following myocardial infarction. So we're really looking at something that we could give, hopefully a pharmacological agent that we could give to a patient who's suffering from a heart attack to protect their heart from damage as much as we can. So during a heart attack, so this rather ropey diagram is supposed to be a heart, and these are supposed to be coronary arteries coming off of it. So if you imagine you've got an occlusion at this point here, That means anything that's downstream of that block of the artery is now at risk from damage during this ischemia. So if you were then to get a spot where you get this necrotic cell death happening, so the bits of tissue are starting to die off, when you relieve that block, these bits of dead tissue is going to be releasing calcium, and there's going to be calcium build up in that tissue because they haven't had the ATP to get rid of that calcium. And that surrounding area, all around here, remains at risk of apoptosis. So this necrotic core kind of spreads out, the infarct size actually gets bigger into that area of potential apoptosis. So stopping the spread of this infarct is really the focus of our current therapies and what we're trying to do in developing new ones. And so really we're trying to cardioprotect the heart, we're trying to limit the spread of this damage. And so how can we really protect the heart from the damage if ischemia is occurring? And there are lots of different ways of doing it, but what's really important is the heart has actually got its own inbuilt mechanisms of protection, and that's commonly just referred to as cardioprotection. And these cardioprotective mechanisms are built in, but we just need to know how to switch them on properly. So cardioprotection is a really broad term that defines any process or intervention that protects the myocardium from damage. Now, cardioprotection can be imparted by short periods of ischemia, by drugs, by anesthetics, and there's all sorts of different mechanisms that have been identified for imparting it, but the mechanisms underlying it are a bit more unclear. It's also important to note that cardioprotection isn't just talking about ischemia and reperfusion injury. So it can also be cardioprotection against arrhythmias, hypertrophy, fibrosis, and also drug toxicity as well. So there's lots of different things that fall under this umbrella term of cardioprotection. But for this talk, cardioprotection is going to mean protection from ischemia and reperfusion damage. So cardioprotection as a concept was really first properly studied back in 1986 when ischemic preconditioning was first identified. Now in this scheme here, you can imagine here you've got normal perfusion in a coronary artery, and then what happens here during this time period is that the artery is blocked by a balloon. So this artery is now made ischemic, or the tissue downstream from this artery is now ischemic. If that occlusion is released and reperfusion happens, you leave it for a while and then you assess how big the infarct is in this tissue. And this work was really inspired by the fact that patients who suffer from chronic angina tend to have smaller infarcts in their main heart attack if they had one following angina. It was as if the heart got used to the small periods of ischemia prior to their big major insult. And so that was replicated using this balloon technology. They put in 3 short periods of ischemia prior to the main insult and assessed the infarct. And what they found is the infarct was much smaller in these ischemically preconditioned animals. So this really became like the gold standard of imparting cardioprotection in the heart. And so the outcome of this was you got a reduced infarct size and an improved functional recovery. But the downside to this is obviously it's not very clinically useful, because in order to impart ischemic preconditioning, you need to know when somebody's going to have their heart attack, which obviously in most cases you don't. So it's not really useful for acute myocardial infarction. It was discovered not long after the identification of ischemic preconditioning that you could do a similar thing, but on the reperfusion phase. So if you re-occluded the artery on the reperfusion, so you had very short periods of reperfusion followed by block again, it was as if the heart was kind of getting used to the reintroduction of the substrates again, so it didn't undergo such substantive reperfusion injury. And obviously this has some better clinical application, because if you were doing a balloon angioplasty, for example, you could, in theory, re-inflate the balloon to re-occlude the vessel to allow this ischemic post-conditioning to occur. But again, this is not something really many clinicians would want to do. You don't want to reintroduce the ischemia. So it's got some clinical use, more so than preconditioning, but it's still not particularly clinically useful. The final major discovery was this idea of remote conditioning. And in this, a limb, such as an arm or a leg, is made ischemic in cycles. And it's done simply by just introducing pressure through a blood pressure cuff. So if you inflate a cuff to around 120 percent of your systolic blood pressure, you'll cut off the blood supply to that arm transiently that can then be released. So if you go through 4 cycles of this ischemia and reperfusion, so 5 minutes of block, 5 minutes of reperfusion, it's not a particularly pleasant experience to have it done to you, but by the end of it, you've released something into your bloodstream that is cardioprotective. And this has been known for quite some time now, but we still don't really know what that substance or those substances are that get released. We're pretty sure it's a humoral mechanism, it's something that's released from that tissue into the blood. So the outcome from this is, again, you get reduced infarct size and improved functional recovery. So potentially it's very clinically useful. And it is under investigation for a number of different disorders. So it's been looked at in terms of stroke and in terms of heart attack. And the downside is that none of these cardioprotective mechanisms seem to work properly in the clinic. And I'll go into a little bit of that later on in the talk. So just summarize where I've got to so far, that cardioprotection is really any process or intervention that protects the myocardium from damage. Now, the opposite of that is obviously cardiotoxicity, any process or intervention that damages the myocardium. And in our lab, we're really interested in both of these aspects. So what is it that protects the heart? What is it that causes the heart damage? So what we really need to know to understand that is really what are the functional characteristics of a cardioprotected cell. What is it about a ventricular myocyte that makes it protected? And the common factor between all of these things seem to be an improved regulation of intracellular calcium. So obviously we all know that calcium is fundamentally important inside any cell, in particular inside a contractile cell, because it's the fundamental part of the contractile pathway. But it also has a lot of other roles within the cells that I've only alluded to with this little diagram here. But what we found over the years is that regulation of intracellular calcium homeostasis is improved in cardioprotected conditions. So if a cell has undergone a cardioprotective stimuli, then it seems to be protected. And the opposite is true, if anything, with cardiotoxicity. If the intracellular calcium is elevated or the homeostasis is disrupted in any way, then that has a toxic effect on the cells. So what's the mechanism underlying the cardioprotection from ischemia then? And this is a 35-year-old research question now that still we don't really have a good answer for. And the problem is really this diagram. And it just shows you the complexity of all the different pathways that are known to cardioprotect a cell. And activation of one of these pathways in some cardiac cells seems to be protective, and it doesn't seem to in others. In some conditions it's protective and some they're not. It's a bit of a mess. There's no one unifying mechanism to all of this. But there are a couple of things that seem to be common to all of these different pathways and all of the different descriptions of cardioprotection in the literature. And those tend to be something to do with PKC and ERK. We have something to do with PKA, particularly involved in the nitric oxide pathways. Also, we seem to have something around PI3 kinase. KATP channels seem to be particularly important, as well as AMP kinase that is meant to act on KATP channels as well. And then finally, something nondescript to do with the mitochondria that seems to preserve calcium and to preserve function. But it's really difficult to see exactly what pathway is kind of the clinching pathway in this cardioprotective process. So really, these are the 3 pathways that most people sort of agree on in the cardioprotection field are the ones that are really important. Protein kinase C, in particular the epsilon isoform, AMP kinase, and then the ATP sensitive potassium channels, the KATP channels. And that's really what we're going to focus on for the rest of this presentation. So the ATP sensitive potassium channel then is a bit of a unique channel in its family. It was first described back in 1983, and every paper ever since on KATP channels says something along the lines of they link metabolic state to electrical excitability. They are exactly as their names suggest, they are an ATP sensitive channel. Under normal physiological conditions of ATP, this channel should be shut. There is an ATP binding site on this pore forming subunit, the KR6, and there are additional ATP binding sites on this SUR protein and also ADP binding sites. So generally, it's thought that this channel is activated by ADP and inhibited by ATP. So under normal physiological conditions, this channel should be shut. and under extreme metabolic stress, it should open. So there are two pore-forming subunits that we know about so far, KR6.1 or KR6.2. KR6.2 is a very well characterized channel. It's found in the pancreas, it's found in skeletal muscle and in cardiac cells as well. It's a really important channel for, indeed, so in the pancreas, it's a really important part of the insulin release process. And in heart and skeletal muscle, it's supposed to respond to really severe exercise to try and reduce cellular damage. So SUR1 is one of the accessory subunits, and SUR2 is the other one. SUR2 has two splice variants, 2A and 2B. So it's a heterooctamer in its structure. You have 4 of these pore-forming subunits come together and 4 of these SUR subunits come together. At the moment, in tissues, we don't think that 6.1 and 6.2 come together to form heteromultimers. We think it's always homotetramers that they form. And that'll become important a little bit later on. So the KR6.2 SUR2A combination is going to be particularly important in this talk because that is the known cardiac sarcolemmal channel. So it's the one that sits on the cell surface. It's also believed to be one of the most abundantly expressed channels in the heart, but that's a little bit odd in many respects because this channel should be blocked by around 25 micromolar of ATP, which in the cell, that should be up to about 4 millimolar. So this channel should never really be open unless you've got absolutely catastrophic ATP depletion. So the other really important channel for this talk is the KR6.1 channel, in particular with SUR2B, is believed to be one of the smooth muscle isoforms. And this channel's slightly different to 6.2, still got the same ATP binding domain in its KR6 subunit, but it doesn't seem to be as sensitive to ATP as 6.2. So KR6.1 in vascular smooth muscle seems to have some sort of really important role in regulating the membrane potential. So it seems to be open all of the time. So really interesting part about KATP channels, I guess, really is the fact that the openers are known to impart cardioprotection and blockers of KATP are known to be cardiotoxic. And that's really where our work came from in terms of trying to figure out what these channels were doing and how they were contributing to cardioprotection. Now, unfortunately, I can't talk about channels without going on a little bit about electrophysiology, so I'm really sorry about this. This will distress some people, but hopefully I'll make what I'm going to talk about relatively simple. I don't need to go into too much detail with this. But very basically, you know that you have, that the cell is essentially a selective barrier allowing ions to be at different concentrations on either side of the membrane. So you've got quite a high potassium concentration inside the cell, a lowish sodium and a very low calcium. And outside the cell, you have low potassium, high sodium and high calcium. So that means there's a concentration gradient. If you open an ion channel, essentially potassium in this case will move out of the cell down its concentration gradient, taking positive charge with it. So that makes the inside more negative. So there's always also a concentration gradient for sodium moving into the cell and calcium moving into the cell as well. If you were to open sodium calcium channels, you get a big movement of sodium calcium into the cell. That brings in positive charge, so that depolarizes the membrane, makes it more positive inside the cell. And that's really all you need to know to understand what it is we're trying to get at with this particular channel. So this here shows you the range of membrane potential that a cardiac cell will go on during its action potential. This point here is really the resting membrane potential. So if you were to open a potassium channel at this point, you would make the membrane potential more negative. You'd call hyperpolarization, and that is a relaxing signal for these cells. If you open a sodium or calcium channel, that's depolarizing signal. It makes the inside of the cell more positive, and that is a contractile signal. So really, there's a constant battle between potassium and sodium and calcium to control the membrane potential of these cells. And that's really easiest visualized in terms of the action potential. So this is a cardiac action potential, cardiac ventricular muscle action potential. So you have a resting membrane potential here, which is controlled by potassium currents. If sodium currents are open, that causes a big depolarizing influx of sodium, so bringing in positive charge into the cell, causes depolarization. You have this plateau phase here, which is mostly where calcium current is active. And you've also got potassium current starting to open during this time to try and bring the membrane potential back down to its negative state. So again, potassium channels are hyperpolarizing, they're inhibitory, they're relaxing to the cells, and sodium and calcium are depolarizing, they're contractile. So what happens to calcium inside the cell during this action potential is something along these lines. So I apologize for my slightly dodgy trace here. I couldn't find a nice one. During the action potential, your calcium rises, and then when the action potential is repolarized, your calcium starts to fall again. And this fall in calcium is due to calcium being pumped out of the cell, either through the PMCA, the plasma membrane calcium ATPase, through the sodium calcium exchanger, or it's being pumped back into the sarcoplasmic reticulum stores. So the KATP channel is really important in this case, because if ATP is low in the cell and this channel opens, what it does is it gives you a really big potassium current, and that big potassium current will actually shorten the action potential because it will increase this repolarizing potassium current. So the more potassium current you've got, the more likely you are to have a short action potential. So if this channel's open to its fullest extent, it will even be able to stop this sodium current depolarizing in the first place, so you won't get contraction. So the ATP sensitive potassium channel in these cells in conditions of severe ischemia will open enough to completely stop contraction happening in these cells. So that's why it's a protective channel. So we believed that in cardioprotected cells, we'd see an activation of this channel to cause this hyperpolarization of the membrane and shortening the action potential to preserve energy. And that was really what our hypothesis would be. So we can measure this directly using a type of patching, a type of electrophysiology called cell attached patch recording. And in this recording, we can push an electrode up against the cell, and basically you can record the channels that are sat in the membrane where this electrode is touching the cell. Now, most channels have got their own individual unique fingerprint, if you like, in terms of their conductance, and we can use this to try and figure out what channels what. So for our particular channel, the KR6.2 channel, in the conditions that we were using, we should have been able to see a single channel current of around 10 picoamps if this channel was open. And if we look on this diagram here, that's exactly what we see. These spikes here, so this is your closed level. These spikes here are single channel openings of that 10 picoamp current, which is the KR6.2 current. But what you can also see in this recording is you've got another little opening here that's above that 10 picoamps. And here you've got another one that's about 5, one here that's about 5, and then another one on top of that. So what it looks like is we've probably got at least 3 channels in this patch. We've got the KR6.2 channel that we're expecting to see. We've got a little channel here that's 5 picoamps, and then you've got 2 of them on top of each other here. So there's probably at least 3 in this patch of recording. So what we hypothesized then is the cardiac KTP current carried by the 6.22A channel activates early following a cardioprotective stimuli to reduce the electrical activity of the cell and protect it from damage. So what we normally see then is during every action potential, calcium would move into the cell, and then be removed here, as I said earlier. But during conditions of ischemia, this would no longer get metabolites coming to the cell, you no longer get ATP production, and so calcium would just increase, and that would cause cellular damage. This KTP channel would normally be blocked under physiologically normal conditions, but it opens in terms of metabolic stress, and so hopefully we'll try and limit the damage that this elevated calcium gives to the cell. So we had this really nice hypothesis, but unfortunately in 2015 we proved ourselves completely wrong. The KR6.2 SUR2A did not seem to be opening early after cardioprotection. In fact, if anything, it seemed to open later. And we did those cell attached recordings, and you can see here, so this is our control conditions, and this is in ischemically preconditioned cells. So these have been through those cycles of pre-ischemia prior to us isolating the cells. So they've been conditioned to be cardioprotected. And what we see is very clearly we've got a much later opening of this channel. So that completely blew it out of the water. So we tried to do some other experiments to see whether we were seeing whether the cells were actually protected or whether there was something weird going on. So we had this really nice little protocol that we've got in the lab where we can simulate ischemia in a dish. So we use a chemical ischemia, if you like, or it's a metabolic inhibition, to cause ATP depletion in the cells, and then we wash that out as reperfusion, and we assess at the end of the protocol how well the cells have survived this. So our cells normally cycle between this low calcium and diastolic phase and a systolic calcium where they're in their contracted phase. So it normally jumps between these two sort of levels of calcium. But on ATP depletion, what happens is the calcium rises further because it can't be removed from the cell. So they actually enter this state of rigor. They shorten down into this horrible little ball of cell that can still recover and can still be functional again, but at that point in time is calcium overloaded and really not in a very happy state. If the calcium remains super high and you give the metabolic substrates back, they enter this state of hypercontraction where they overcontract and literally, as I said, pull holes in themselves. So what happens during this protocol is the ATP depletes during time, and we see that the KATP channels open, which causes the cells to fail to contract. As I said, the opening of that channel stops those cells contracting. You get elevated calcium because that ATP is depleted. In a proportion of cells on the reperfusion, you'll get hypercontracture, which is equivalent to the reperfusion injury I talked about in the whole heart. And then on reperfusion at the end, towards the end, the cells start to recover their calcium homeostasis again and recover contractile function. And our cells in a dish look something like this during the protocol. So we've got these nice, long, healthy looking cells here. All these round ones are the dead ones, we ignore those. And then at the end of the poison, these cells have shortened down into this rigor state they're described here. And at the end of the simulated ischemia, unfortunately, because our cells aren't electrically linked, aren't physically linked to one another, they never regain their long morphology that they started off with. In the heart, they would have the blood pushing on the cells and pulling them back out again, but that doesn't happen in this protocol because they're not physically coupled to one another. But we can still see them contracting at the end, and we measure that contraction. And then finally, we use trypan blue to label those cells that are dead versus those that are alive. So you can see here, this one is in rigor, but still, I can tell you that one was contractile. But here you've got one that was completely dead, and you can see it's got membrane blebs coming off the side of it, so it's really not a very healthy cell. And the data from that protocol really backed up what we were thinking, because what we found in control conditions is that we got a contractile failure time of somewhere at around 6 minutes into the protocol, but that was really prolonged in ischemically preconditioned or adenosine treated cells. So adenosine is thought to be part of the ischemic preconditioning pathway. So what we found is that we had a delay in this time to contractile failure, so a delay in the time to activation of the KATP current. So even our cellular data, our whole cell data, not the electrophysiology stuff, was telling us that the KATP channel was activating later in this. And what we concluded in this paper essentially was that KR6.2 SUR2A was actually an ATP sensor rather than a protector of the immediate cardioprotection that we were seeing. So these cells were able to preserve their ATP for longer during ischemia, but it wasn't due to the activity of this channel. But that kind of left us with a bit of an awkward question, because why do pharmacological activators of KATP impart protection when the channel in the heart didn't actually activate? So it was quite a difficult bit to us to try and figure out what was going on. But then we sort of looked more carefully at the data following on from this and following on from a series of revelatory experiments in the lab, where we actually noticed that it was the data, the reason was probably hidden in our own paper. So if you look here, you can see that there's actually quite a lot of these spikes going, although this is quite a low resolution recording, just to try and show the whole trace. If you look at the high resolution recordings, you can actually see these little half step openings that we would see. And we saw these really regularly, and even in that first diagram I showed you here for the cell attached recording, you can see those little half step openings when KR6.2 was opening. So this second channel in this recording seemed to be present all the time, but it wasn't the standard KATP channel, and it also wasn't the inward rectifier channel that sets the resting potential in these cells. So for this paper, we kind of ignored it, but we probably really shouldn't have. Well, perhaps we should, because it's now given us this future work, and we've got 2 papers out of it. So what we found is that this small KATP-like current that we thought it was, but not only did it show openings all of the time, even at rest, but it was also sensitive to some of the classic KATP channel blockers. So this little channel was actually sensitive to glibenclamide, which is the standard KATP channel blocker that people use. But it was also sensitive to kind of a newish compound, this PNU 37883A, nice, handy, easy to name thing, which I'll call PNU from now on. It's a selective KR6.1 pore blocking drug. So it binds to a region of the pore inside the channel rather than binding to the receptor like the other drugs do. So it actually physically blocks the pore. So it's quite a different drug. What was also interesting about this channel is that if we applied an activating compound, panacedyl, we saw this channel's activity increased. So what we realized is that we had something that was looking like a KTP channel, but wasn't the standard cardiac KTP channel. And this channel was showing constitutive activity, i.e. it was active all of the time. And that was really quite like what happened in the vascular smooth muscle, that this channel's always open there. So this KR6.1 like channel was showing the same kind of properties that the smooth muscle channel shows. So just to try and convince ourselves that it really was the KR6.1 versus KR6.2 thing that we thought it was, we expressed KR6.1 with SUR2A in a HEC293 cell. We measured the current in the same kind of way. And in our cardiac cells, we saw something which gave us this kind of opening. And in the HEC cells, we saw this. And these channels really did map in terms of their conductance. So this is the conductance for the channel in both systems. So the dotted lines are the HEC cells and the squares are the cardiomyocytes. And the same if we did KR6.2, we got virtually identical channel properties. So we really did think that we were seeing two distinct KATP channels. Our final kind of really important bit of evidence, I think, is the fact that in our KR6.1 knockout animals, where we went to Andy Tinker's group in London to do these experiments, we couldn't see this channel being present in these cells. So it seemed to be absent, which again gave us a really good indication this was actually the channel we were looking at. So if the channel is active under normal physiological conditions, does that mean it plays a role in normal physiology that we've perhaps overlooked previously? And we actually think it probably does. So this experiment here is looking at the cardiac action potential from a rat ventricular myocyte. Now it's very different to a human action potential. It's much more triangular in shape. You don't get a long prolonged plateau phase like you do in the human action potentials. But what you do see, if you use this drug, this 3 micromolar PNU compound, you actually get a prolongation of the action potential longer than it normally is, which is what we anticipated would happen if this channel was there all the time. We thought if this channel was active all of the time, it would be having a role in shortening the action potential and fine-tuning it. And Richard Barrett-Jolly helped us with a simulation of the human ventricular action potential as well. So we added this channel into this human simulated action potential and then knocked it out again. And we showed that we got a prolongation of that action potential by removing that channel. So the modeling really did go with what we were seeing really nicely. So then we looked again to sort of further confirm this was having a physiological functional effect on our cells. We showed that if we looked at intracellular calcium, that the calcium accumulation during each contractile cycle was larger in the presence of this blocking drug. So blocking this channel was causing an increase in calcium. That was giving us larger contractions, as you might expect. If you've got more calcium in the cell, you'd expect them to give bigger contractions. And then in our simulated ischemia and reperfusion model, it was actually shortening the time to contractile failure, suggesting they were running out of ATP more rapidly. It was reducing the contractile recovery in the cells and reducing cell survival. So blocking of this tiny little channel was really having quite a profound effect on the outcome of this ischemic conditioning. The final bit of evidence for this channel having a negative effect if you block it came from some whole heart experiments where we did some coronary ligation. And in these experiments, so these are rat hearts that are on a Langendorf cannula. And what you can see, you might not be able to see it very clearly here, but there's a coronary artery that runs down the length there. This is the left anterior descending coronary artery. And if we block that for 40 minutes, it says 30 minutes, there's actually 40 minutes, and then release it for a reperfusion for 3 hours, we can actually measure the size of the damage that's actually caused to the heart during that coronary artery block. So what we do is we tie this artery back up again, and then we perfuse through into the heart this Evans stain blue. Now this will go into all of the bits of the heart that weren't supplied by that one coronary artery. So anywhere that that artery supplied will actually stay red. And what you see, if you look at some of the example slices we get from that, if we do slices right the way through the heart, you can see that you've got these big areas of red and these little bits of blue that weren't affected by that coronary artery. So what those different areas show is you've got a big hole there, which is our left ventricle. You can't always see it very clearly, but there is a right ventricle hole. So the right ventricle is always a lot less muscular than the left ventricle. And then what you see is an area at red, which is your area at risk. And that's done by staining the cells with TTC. And then finally, you'll see an area of white with the stain here that indicates your area of infarct. So we can quite reliably use image J then to calculate how much of this area at risk was actually damaged during that simulated heart attack. And what we find under normal conditions, we get somewhere between 30 to 40% of your tissue being damaged. But if you block the KR6.1 channel selectively, you get a much larger infarct size. So it's really causing a lot more damage to these cells. So selective inhibition of KR6.1 worsens the outcome from this ischemic insult. Conversely, if you apply panacetyl, which is an activator of the compound, and if we use it at 10 micromolar, we selectively activate KR6.1, then we actually get protection. So block of 6.1 is cardiotoxic, enhancing 6.1 activity is cardioprotective. Now, ischemic preconditioning, we know, protects cardiac cells from damage, but glibenclamide and other non-selective KATP channel blockers abolish that cardioprotection afforded by ischemic preconditioning. So we wondered, does ischemic preconditioning actually activate KR6.1 in ventricular myocytes? And what's really cool for us is the short answer to that was yes, it does. So if we do these cell attached patch recordings again, so this is our openings and control conditions, and after ischemic preconditioning, the activity of this channel has gone through the roof. It absolutely goes mad. So KR6.1 activity is significantly higher in ischemically preconditioned cardiac myocytes. And again, here we've shown that this higher open probability can be blocked by the selective compound and the non-selective KATP channel blocker. So it really is having a protective effect on the cells. It gets a little bit more complicated because what we seem to see with our data is that we've got some sort of bimodal distribution of our open probability in control conditions. So we have an area here where we've got a lot of these cells where they fall in the open probability here. Then we get this second peak here. But what's kind of nice for us is that with the IPC cells, the ischemically preconditioned cells, all of them seem to be in this second region of our NPO. And that kind of suggests to us that the higher the open probability of the channel, the more protected the cells are. And in our ischemia assays, we tend to find that about 30% of cells survive our ischemia and the rest kind of fall into a not so surviving it so well group. So we're suggesting that perhaps in these cells that do survive our ischemia and control conditions, they're actually in this, potentially in this ischemically preconditioned group. So again, we've shown that our ischemically preconditioned cells show a larger contractile recovery. They've got this delay in their time to contractile failure, suggesting that their ATP depletion is slower, and they've got this increased cell survival. And all of those can be reversed by selectively blocking that channel. And again, in ischemically preconditioned whole hearts, we can show that PNU reverses that protection. So we think we've got some really nice data now suggesting that we've got some sort of mechanism coming through of how KR6.2 might be protecting cells. So firstly, we've got KR6.1 activity being increased. That shortens the cardiac action potential duration, gives you decreased time for calcium accumulation. That reduced calcium accumulation reduces the energy required to kick that calcium back out of the cell. So your ATP concentrations are preserved for longer inside the cell. That means it takes longer for the KR6.2 to respond to that ATP depletion. And it means at the end that you get a delayed time to ATP depletion. The cell doesn't load as much with calcium, and so the reperfusion injury is limited. And just a little bit more evidence for this. This is a plot of our KR6.1 open probability against the time to KR6.2 activation, so the time at which ATP is depleted enough for that channel to open, and the contractile recovery, so how protected those cells were. And what we see is a really nice correlation there between the open probability being low, and you've got a low contractile recovery and a short time to ATP depletion. With a high open probability, you've got a high contractile recovery and a high time, a delayed time to KR6.2 activity. So we think it's a really nice story that we've got here. So additional evidence for this being that cardioprotection imparted by ischemia is only transient, so it only actually lasts for about 12 hours in cells. And if you look at our enhanced contractile recovery, we can measure that during the day. So the longer it is from the time that we isolated the cells or imparted that protection, the smaller that protection is. And again, if we look at the open probability of the channel, the further away from the ischemically preconditioned, the lower the open probability is. So it seems that the contractile recovery and the enhanced open probability of the channel is waning during the day. If we look at these preconditioning as well, so I said about short periods of preconditioning protect from a longer period of ischemia. If we do an experiment to look at that, we can show it in real time. If we give metabolic inhibition, so we activate the 6.2 channel, and then look again at the 6.1 open probability, it's much higher following a first round of conditioning, a first round of metabolic inhibition. So we're actually seeing in real time an increase in this channel's activity. So we think that maybe KTP is our missing link between the KTP pharmacology and the channel activity. So KR6.1's got this really important and potentially previously overlooked roles. But we think this has also got some other implications as well. So for example, rosiglitazone is a compound that was given as part of the treatment for diabetes, and it was a PPAR gamma agonist. I think I may have got that wrong, but it was found that it was increasing the likelihood of deaths from ischemic insults in some circumstances with a really unclear mechanism. So it was withdrawn from the market. But it's also been shown that this channel has been shown to block KR6.1 with no effect on the other KTP current. So we did a concentration response curve for rosiglitazone, showing we got a reasonable response and inhibition of this channel, but the other compound pioglitazone didn't have the same effect on the channel. In fact, if anything, the open probability was slightly higher. And in our cellular assay again, we found that rosiglitazone actually worsened the outcome for the cells, whereas pioglitazone had no effect on it. So again, a different drug blocking KR6.1 potentially had this negative effect on the cells. So something that we wondered, should KR6.1 be considered by drug companies who are looking at cardiotoxicity screens for new compounds, because it's not known to be in the myocardium as yet. So hopefully we're going to give some information as to whether this channel should be considered as well. So we're trying to send this paper to circulation research, so it's under review at the moment. And so in answer to the question that I posed as the title, I think yes, but it's a very complex situation. We think that it plays a role, but not the KTP channels, perhaps not in the way that they were previously anticipated to play a role. These vascular-like channels are expressed at the cell surface in these cardiac cells, and they're playing a role of fine-tuning calcium. Their activities increase following stimuli. Their activity correlates with the level of protection seen in the myocardium as well. But we also think that the KR6.2 are a really important part of that cardioprotection process, because they're there to respond to a catastrophic depletion of ATP. So we're not saying they're not involved in protection, but they're not involved in this early protection. And then, could KR6.1 be a pharmacological therapeutic target? Well, we kind of hope so, but it's really important to note at this stage that cardioprotection doesn't actually work in man, which is really frustrating for everybody. So very briefly, I'm just going to finish off by saying why we think cardioprotection doesn't work. There's really good in vitro evidence that it works. It's fantastic in cells, works brilliantly every time. Brilliant in vivo efficacy as well. So you can do animal models and you can show that cardioprotection works. But this just doesn't translate to the clinic. And we suspect that it might be something to do with how animal experiments are done versus what happens in the clinic. So we published a paper a while ago now showing that high glucose activates protein kinase C to cause cardiotoxicity. And without wishing to go into this paper too much, this was something that was quite interesting to us, because we managed to get a bit of clinical data in this as well. And we think that stress causes high glucose, which causes PKC activation, which causes cardiotoxicity. Now, animals are always really carefully sedated in all of these preclinical studies. So they're sedated, they're under pain management strategies prior to any modeling of heart attacks that they have. A human who has a heart attack is very aware they're having it and is obviously going to be extremely stressed by that. And the stress response releases glucose into the system, which could therefore cause negative effects on activating PKC and imparting cardiotoxicity. And what's also really clear is that high levels of glucose abolish cardioprotection in a number of different scenarios. Just to go through a little bit of the data we had here in the humans, we found that I think it was 47, actually I've written 49 here, percent of non-diabetic patients had a blood glucose above 8 millimolar when they went into hospital on first identification of their heart attack. So that kind of would suggest to us that that's going to cause some negative effects on cardioprotection, because in a previous paper we did, we looked at how glucose abolishes ischemic preconditioning. And by the time you get to 20 millimolar glucose, your ischemic preconditioning is going to be completely wiped out. So any cardioprotection you had is wiped out by glucose. And in subsequent studies and stuff that we're working on now in the lab, we've shown that most protective stimuli are actually wiped out by having high glucose. So we suspect that clinical trials that have obviously used high glucose, have obviously used patient studies, whatever, have not really accounted for the fact that some of these patients have got hyperglycemia on admission to the clinic. And so therefore that's going to cause, we think, a disruption of their results. So it's something that we really want to consider looking at in the future, is previous clinical data, and can you look at the blood glucose of the patients at the time and see if you can separate them out based on that. So we think that targeting KATP channels directly, rather than trying to go through all this cell signaling, might be a novel mechanism that we can use for treating cells and giving cardioprotection, because also in Sean's paper back in 2015, we showed that all of these other mechanisms of cardioprotection were dependent on this cell signaling PKC, but panacetyl direct activation wasn't. So I'm going to leave the data there. I'm just going to very quickly introduce my lab. So in case you see any of us wandering around, we look a bit lost in our new institute. So this is Sean and Simona and Abra, my PhD students. This is Mohammed, another PhD student who's based in Leicester at the moment, but hoping to come to Liverpool. And this is Fada, another my PhD student who's recently joined us. And the funny thing about this photo is, as I said here, they moved with me from Leicester in February 2018. It was 4 degrees, howling wind, raining. They wondered what on earth they'd done coming to Liverpool, but thankfully they all seem to be quite pleased with our choice. So just want to leave this slide up to say thank you to all those who have helped with this work, and thank you very much for listening.
Well, Richard, thank you very much. This is all very outside my comfort zone, but I understand it a lot more than I thought I ever would. So thank you for going through it in that way, because I think that personally it really helped me to be talked through it, and to understand sort of a little bit more about this sort of what it is you're actually measuring in these experiments. So Richard, you've got a couple of questions. Do you want to, I think you've only sent them to me, so I don't suppose Richard can see them. Do you want to talk about your questions, Richard Barrett-Jolly?
Okay, Richard. Yeah, thank you, Richard. I enjoyed that a lot, of course. So I had a couple of questions on this. The first is kind of a little bit peripheral. You're showing this cardioprotective pathway. What actual pathway is that? Is that a KEGG pathway, or is it one that you sort of manually created? Or
I can't claim.
You showed it a couple of times. I expect it's KEGG or something, but I just it's interesting, and I thought it might be interesting to look at some of our transcriptomic data with reference to that.
Oh wow, that's right. So this is kind of from a review that kind of goes through all the different pathways identified. So it's not, I can't claim this is mine at all. This is something I've nabbed from somebody else.
Right, okay. Well, I might come and grab that reference from you at some point then, thanks. And the other one was a bit more sort of a bit of a bigger question, really. You've kind of shot yourself at the end by kind of going, doesn't work in the clinic. And I kind of buy the arguments about, well, I understood the arguments about hyperglycemia and the glucose, the PKC changes, et cetera, but what actual drugs have they used in what kind of clinical trials have they used in people for this? Have they, I mean, they tried KIR, I mean, specific activators of KIR 6.1? Or?
As far as I'm aware, no, they haven't, because things like panacetyl, which would be the obvious one, I think is not selective enough to be used.
No, no, not at all.
I think nicorandil was tried at one point, but that has huge amounts of other effects as well. It's nitric oxide donating as well. So I don't think there's been any KR6 particularly used in clinical trials as yet. Certainly adenozine's been tried. It's remote ischemic conditioning is ongoing in all sorts of clinical trials, and again, even that's failing in clinical trials. But again, we found when in Mohammed's experiments, when he's looking particularly at hypertrophy as well, but he's found that high glucose blocks the protective effect of his conditioned serum against hypertrophy as well. So it seems that glucose is having a lot of effects on these potentially protective pathways that is just blocking their activity, which is why we're really interested in the idea of going directly to the channel to protect rather than trying to go through the signaling.
Yeah. So I mean, it's possible that it would work in humans if you found the right selective enough drug, isn't it?
So there is a compound out there in China, I think, is iptacalim, I believe, is the one that people have tried using, which is supposed to be selective to 6.1, but we've not been able to get hold of it. We have tried a couple of times.
Okay.
So, Richard, would it make any difference if you delivered the drug directly to the reperfused or area? So, for example, if you actually had it coming off the stent or something, so that it wasn't, that it was really concentrated in the cells that you actually wanted to get it to.
So that's one of the things that we've been really interested in this as a whole, because technically, because the heart is a syncytium, if you affect one region with causing changes in electrical activity, you should technically be able to affect all of it, because the hyperpolarizing effect of a channel at one area should technically affect another area as well. So you may not need to get it into the area that's damaged in particular. But yes, in terms of drug-eluting stent, that would be really helpful, possibly. The downside is that the channel is also expressed in the vasculature. So whether it would affect the vasculature more than the cardiac cells, we don't know, on whether how good tissue penetration would be.
Okay.
But no, it's a really interesting point. It's something we've been thinking about for ages with this project, of how you spin that as a potential clinical tool.
Yeah. Good. Any other questions? Anyone has questions, please add them to the chat, or if you just want to ask them, please unmute yourself and ask. It must have raised more questions. No?
Apparently not. Oh well.
Well, it was incredible. It was very clear, actually, Richard. I mean, I think to me, it is an area which perhaps a lot of us aren't as familiar with as in other areas. It's not, we haven't had this within our, you not joining our institute is a real advantage to us, because it's bringing in a whole new area that is really useful. So I think people need to go away probably and think about it a bit more, and hopefully they'll come back to you with some questions. But it really was very clear. Yeah, Graham says, Graham Kemp says, yes, indeed, excellently clear. So
Well, thank you very much.
very nice. Thank you very much. And I'm, giving a seminar to your own screen is difficult, as we all know. So, and hopefully, I'm sure this will lead to other conversations within the Institute over the coming months. But everyone, thank you very much then, and particularly thank you to Richard.
Thank you. Bye-bye.